well, thank you everyone for being here at our PhUnC conference. Thank you especially to all the people that traveled away to be here. It's pretty cool. So our first speaker is Ahmad Mahmood, and he's going to be presenting Organelle with License to Kill.
Yeah. So my name's Ahmad, as I don't know your name. Colin. Thank you for having me here. So my name is Ahmad. I'm from McMaster University, and I had the pleasure to work with Dr. Cecile Freyden in an experimental biophysics lab over the summer, and with her PhD candidate, Marcus Rose. So the title of my talk is 007 Mitochondria and Organelle with a License to Kill. So my talk mainly revolves around a specific type of cell death known as apoptosis. And apoptosis, Latin for falling off, is a series of structural sequenced events that leads to the ultimate death of the cell. And this cell death is marked by morphological changes that we see when we look under a microscope at different cells. And there are two different ways that this cell death can occur, through an extrinsic pathway or an intrinsic pathway. An example of an extrinsic pathway is, say for example, a cell has damaged DNA. It's not favorable for that cell to reproduce itself or to replicate for that entire biological system. So it will get signals external to the cell, and it will tell the cell to die. So on the left-hand side, what we see is a mouse paw. And what we know in even in most vertebrates and in humans, that when we're in the fetus or in embryo, we have webbing between our fingers. And this webbing eventually, in most cases, degrades away. And this webbing and this degradation is due to apoptosis. So how is apoptosis regulated? So the regulation of apoptosis is primarily controlled by a family of proteins known as the BCL2 family of proteins. And they're a family because they share what are known as the BH1 through 4 homology domains. And this simply means that they share a similar structure. And this structure is shown through these colored boxes. So you see BH1, BH2, and BH4. And the primary focus of my talk are on two BCL2 family proteins known as BAX and BID. BAX is a pro-apoptotic member, and by pro-apoptotic, it means that it promotes the death of the cell. And BID is a BH3 only protein, so it only has the BH3 domain. And the function of that protein is to regulate the activity of BAX. Throughout my presentation, you will see TBID up there. Not BID, but I will say BID or TBID. By TBID, I mean truncated BID, and that simply means that it has been in an activated form, that it has been cleaved into two pieces. One, TBID is slightly larger than smaller than the whole BID protein. So what happens when it is regulated? So in a healthy cell, so this is the mitochondria down here, and we have an outer membrane and an inner membrane of the mitochondria, as we know due to the endosymbiosis theory. And between these membrane spaces is known as the intermembrane space. So this is where the mitochondria plays a huge role. It's not just the powerhouse of the cell. So in a healthy cell, BAX just transiently interacts with the outer membrane. It doesn't poke holes or it does anything. It just comes by, and then it goes back into the cytosol. And everything's good, it's a healthy cell. But what if it receives a death signal, either through an extrinsic or those intrinsic pathways? What happens is that BID, or TBID in this case here, it translocates to the mitochondrial outer membrane. And what this does is that it recruits BAX proteins to the membrane and activates them. And through this activation, it recruits more BAX proteins to the outer mitochondrial membrane. And this leads to the initialization step of apoptosis, known as the permeabilization of the mitochondrial outer membrane. And this permeabilization is due to the fact that once these BAX proteins have accumulated, they can oligomerize. And oligomerization is simply the formation of a slightly long chain, about 30 units or so, and a pore can form on the mitochondrial outer membrane, and this allows a protein known as cytochrome C to travel from the intermembrane space out into the cytosol, and through a series of a lot of cellular activity, it leads to the death of the cell. But what happens when it isn't regulated? And that's what leads to the motivation of the research at hand, is that when there's too much apoptosis, it can lead to autoimmune diseases or stroke, and when there's not enough, it could lead to cancer. And that makes sense, because if our cells aren't dying, they're just accumulating and accumulating. And this apoptosis is just completely regulated by the BCL2 family protein. So how do we study this BID-BAX interaction at McMaster? What we do is we target BID and BAX proteins through fluorophores, so we label each of them, and then we irradiate them with a certain wavelength of light. So here, BID proteins are most often labeled with a green fluorescing fluorophore. And here on the left-hand side, we see we make these two-dimensional, well, three-dimensional, but effectively two-dimensional synthetic mitochondrial-like outer membranes through different lipid compositions. And what we do is that we flush them with proteins, and we scan them with scanning confocal microscopy. And scanning confocal microscopy is what I like to call reading a book. What it does is that it kind of reads from left to right in a horizontal fashion, like we read books. And what we see is that if the proteins are very mobile, we see them as streaks. And if they're very immobile, we see them as spots. So here, green laser, we see this kind of as a spot, but we see some things that are more streak-like. And this means that the streaks are low interacting with the membrane, whereas spots are embedded within the membrane. So what we do is that we label BID with a green fluorophore, and BAX only glows when we irradiate it with red light. So what we do is that we correlate spots and streaks against each other. And what you see is, for example, in this case, that the dimensions of this image are 10 micrometers by 10 micrometers, which is very, very small. So these spots are close enough together that we say that they are interacting with one another, and we say that some kind of complex has formed. So what we want to do is that because these images are two-dimensional, we're not getting a three-dimensional understanding of what's actually going on. What we want is a three-dimensional understanding of the protein dynamics. So that led to a research question, is that can we determine some kind of algebraic relationship where we can take our two-dimensional protein dynamics that we get from our images, and using a little bit of modeling, mathematical modeling, can we get three-dimensional protein dynamics, which what we're really interested in. And what we do is that we introduce variables from our experimental setup. So the experimental setup includes the different proteins, or sorry, the different lipids that we would use to create those synthetic bilayer membranes. So let's introduce a very simple model at first, which I like to call the mystery box model. What we do is that we introduce BID and BAX, and we don't know what's going on, we don't know what type of interaction's happening, but we do know what we get out of this interaction, what we call a heterodimer, or a BID-BAX complex. And this reaction can be represented mathematically, or we're more used to seeing in a chemistry class, is a reaction equation where BID plus BAX yields a complex, and we can represent this with what's known as, in this case, a 3D dissociation constant, which is simply a ratio of the amount of free BID and free BAX that exist at equilibrium, the concentrations, divided by the concentrations of the complex itself at the very end at equilibrium. So the units of these are in some kind of molarity, which is a three-dimensional quantity. But we can make this model a little bit more complex, and we can introduce a membrane into our mystery box model, where first the BID and BAX proteins have to interact on the membrane, and then they can form a complex on the membrane. So this can be shown graphically as something like this, where the BID and BAX come from the cytosol, they travel, translocate to the mitochondrial outer membrane, and then they can form a complex on the membrane. And we can develop a set of equations for each of these motions. So let's focus on the BID. So what we do is that BID translocates from the cytosol to the membrane, and we can describe this by what's known as a partition coefficient. And that is a description of how much BID we will find in the cytosol versus how much we'll find at the membrane at equilibrium, and it's a ratio of those two quantities, those concentrations. So likewise, we can do something for BAX. where we introduce another partition coefficient, where how much will be found in the cytosol versus how much will be found at the membrane at equilibrium. So now, once they're at the membrane, they can finally form a complex, as before. So this equation looks very familiar. However, the only difference is that this is two-dimensional, as it exists on a two-dimensional membrane. So here we have our free bid, our free backs, and then the complex, and the ratio of those at equilibrium. So without going through a lot of the math, we can skip over that because it's a physics conference. What I wanted to do was that I wanted to equate the simple model and our complex model. So our simple model yielded us a system of 3 equations, whereas our complex model yielded us a system of 5 equations. And what we wanted to do was to find an expression for our 3-dimensional dissociation constant as a function of our 2-dimensional dissociation constant. So what we can do is isolate for our 3-dimensional dissociation constant using a lot of mathematics, because I didn't want to do it by hand. And what we can do is that we will get an expression, but what's important to note is that these partition coefficients are fixed. So what we have is a function that's dependent linearly on another variable. So from math class, we know that if we were to plot something like this, we should get a line. And that's exactly what we got. And I was very happy with that. So what we can do is we can test this model a little bit. We can test this theory. So from experiment that was done in our group a couple years ago was that we found that the partition coefficient of bid is around 10 to the negative 4. And that was from Shemstein 2015. And what we know one point on this graph that has to exist is that this line has to go through the origin every time, because if there's no proteins, then there can't be any dynamics. So that is one point that's given. Another point that we have from experiment is that when our 2-dimensional KD is about 2 per micrometer squared, our 3-dimensional KD should be about 20 nanomolars. So now we have 2 points, and we want to fit a line through it. We can do that. And I can do that by adjusting the slider. And that gives us a partition coefficient for backs, which we don't have experimentally, of about 3 times 10 to the negative 3. And that is actually a good first order approximation for our model, in that this number tells us that backs like to spend more time in the cytosol, and that has been shown experimentally to be true as well, because this number is larger than this, which means that bid likes to spend more time at the membrane. So, knowing that this model is a good first approximation, it's not perfect, which we can see in our conclusions is that we can create these synthetic membranes, which are biomimetic, which means that they resemble the mitochondrial outer membrane. And we can analytically model protein-protein dynamics and protein-membrane dynamics. And we can extract information like partition coefficients, which will tell us how much do we expect to find at equilibrium in the cytosol or at the membrane. And for future work, what we can do is increase the model complexity for oligomeric reactions, which I've been working on a little bit, where instead of just a heterodimer forming where bid plus backs, I stated earlier that backs proteins like to oligomerize to form these pores. What we want to do is be able to model for multiple chains, like a bid plus backs, something like that. An experiment has shown that the most likely form of a Bax protein when it forms a pore is something like a hexamer or a former or a tetramer. And what we can do is that we can explore more than 2 protein interactions with something called rule-based modeling. And apoptosis has many, many proteins involved in it, and we just looked at 2, which are the key players in mitochondrial outer membrane permeabilization. So with that, I conclude my presentation. So I'd be happy to take any questions. Yes?
Very nice talk.
Thank you.
Can you describe the picture better for me in terms of the process of apoptosis?
Sure.
Whether the backs will arrive, and the T-bit will also arrive, and to the mitochondria, and they both will be increasing in concentration.
Yes. So if I can just flip back. It's a very nice this picture I thought was the most simplistic one that I found from Luo in 2014. So what BID does is that once it gets that signal, it will translocate, and BACS initially will not permeabilize the membrane because it hasn't been activated. So BID has alpha helices, which will insert into a domain onto BACS, and it'll kind of unlatch. And these alpha helices can then insert itself into the membrane. And that can't be done without this BID activation. And once BID is there, it can recruit more and more to BACS. So it's more likely for BACS to come to the membrane once it's received this death signal.
So once they get it, would it ever be able to reverse that?
Yes. So one of these proteins that I haven't talked about is BCL-XL. So if I go back one more, BCL-XL is a pro-survival member. So which means that it wants to prevent apoptosis. So BCL-XL is kind of like a shuttle which carries the BACS back to the cytosol. So what we want to do is that if we can understand these cellular processes, we can accelerate or decelerate this apoptotic process, and that'll be able to target certain diseases like cancer on the cellular level instead of the macroscopic level.
Thank you.
Yep. Anything else? Yes?
So you just need one of these BID proteins to collect many of the BACS proteins to the membrane, or?
So obviously this is a very simple model where you have a lot of BID proteins that are all over the mitochondrial membrane. What I read was that you don't need a lot to be able to recruit more and more BACS. So I like to think of like a catalytic reaction where once it sees that a BID has traveled to the membrane, then BACS is like, okay, let's start moving there. Yeah.
Thank you very much.
Great.